Welcome to BioBits and Bytes.
Great to be here.
Today, we're diving into something really fascinating. It's where tiny, tiny technology meets a huge challenge, Alzheimer's disease.
Exactly. We've been looking at some cutting-edge research, haven't we? All about how nanotechnology might give us new tools to tackle it.
Right. And our mission here is to break down this complex science so you, the learner, can really get a handle on it without feeling overwhelmed. Think of it as getting the inside track.
We're focusing specifically on nanoparticles, these incredibly small particles. The idea is, how can we design them to actually go after Alzheimer's?
And the research suggests some really interesting possibilities, like delivering drugs right to the brain.
Potentially breaking down those harmful protein clumps we see in Alzheimer's.
And even getting past the brain's defense system, the blood-brain barrier.
Which is a huge deal in itself. We'll be looking particularly at Donepezil and PLGA nanoparticles as key examples from the studies.
Okay, perfect. Let's start with the basics then. Alzheimer's disease. We hear the name a lot, but could you maybe define it clearly for us? What exactly is happening?
Sure. So Alzheimer's is a progressive neurodegenerative disease. That means it gets worse over time, and unfortunately, right now, it's irreversible.
Progressive, okay.
Essentially, the nerve cells in the brain, the neurons, they become damaged and die off. This leads to that decline in cognitive functions, memory, thinking, reasoning, all those things.
And current treatments, what's the main approach there?
Well, current treatments are mostly symptomatic. They focus on trying to improve how the remaining nerve cells communicate, boosting that neural transmission.
Improving the signals. And one common way to do that is using cholinesterase inhibitors, or TEIs.
That's right. TEIs are a major class of drugs used for Alzheimer's symptoms. Could you explain those a bit more?
Absolutely. So there's a chemical messenger in the brain called acetylcholine, really important for memory and learning. Cholinesterase is an enzyme that naturally breaks down this acetylcholine. So these inhibitors, the TEIs, they block that enzyme.
So they stop the breakdown.
Exactly. They slow down the breakdown, which means there's more acetylcholine available for the brain cells to use, hopefully improving that communication.
Makes sense. And this brings us to Donepezil, which features heavily in the research we looked at. What is Donepezil specifically?
Right. Donepezil is a key example of a TEI. It's specifically known as a reversible and non-competitive TEI.
Reversible and non-competitive. Can you unpack that for us?
Sure. Reversible means its effect on the enzyme isn't permanent. It blocks it temporarily. Non-competitive describes how it interacts. It doesn't directly fight acetylcholine for the same spot on the enzyme.
Got it. And it's used clinically.
Yes, it's used clinically for palliative treatment, so it helps manage the symptoms, improve quality of life, but it doesn't stop the underlying disease progression.
A crucial distinction. Okay, so we have this disease and drugs like Donepezil that help manage symptoms. Now, where does nanotechnology come in, this nanoparticle approach?
This is where it gets really interesting. The core idea is using these ultra-small nanoparticles as, delivery vehicles.
Tiny taxis, almost.
Exactly. Tiny taxis to carry drugs like Donepezil, specifically aiming to get them into the brain where they're needed most.
And one material that keeps popping up is PLGA. What is PLGA and why is it suited for this?
PLGA. It stands for polylactic coglycolic acid. It's a type of polymer, a long-chain molecule.
Okay.
It's actually a copolymer, meaning it's built from two different building blocks, lactic acid and glycolic acid. They're linked together.
And you can vary how much of each.
Precisely. Scientists can change the ratio of lactic to glycolic acid, which lets them fine-tune the properties of the final PLGA material. Very versatile.
And I gather lactic acid itself has different forms.
It does, yeah. There's a D form and an L form. They're like mirror images. And you can make PLGA with an equal mix called poly-D, a lactic coglycolic acid.
Okay, so it's customizable. But what makes it good for use inside the body?
Three key things, really. Biocompatibility, biosafety, and biodegradability.
Right, biocompatible means.
It generally doesn't cause a bad reaction in the body. Biosafety means it's considered safe for biological systems.
And biodegradable.
That's a big one. It means the PLGA material breaks down naturally inside the body over time.
Into harmless stuff.
Exactly. Into non-toxic byproducts the body can just get rid of. So the delivery vehicle dissolves after it does its job.
Very neat. So the research looked at loading Donepezil into PLGA nanoparticles, but specifically PLGA BPEG nanoparticles.
Yeah.
What does that BPEG add?
Good question. PLGA BPEG, the BP means it's a block copolymer. You've got a block of PLGA attached to a block of another polymer, polyethylene glycol or PEG.
PEG, I've heard of that. It's
Okay.
used in other medical contexts, right?
Yeah. It is. Attaching PEG to the surface of nanoparticles is a really common strategy. PEG likes water, it's hydrophilic.
Okay.
So it forms this sort of water layer around the nanoparticle, and that layer acts almost like a stealth cloak.
Hiding it from the immune system?
Pretty much. It helps the nanoparticle avoid being quickly detected and cleared by the body's defenses so it can circulate longer and hopefully reach its target.
Clever. So these stealthy Donepezil nanoparticles, the study looked at two key things. Could they mess with those amyloid beta fibrils? And could they cross the blood-brain barrier? Let's take those one by one. Amyloid beta fibrils.
Yes, afibrils. These are basically abnormal clumps or tangles of protein that build up in the brain in Alzheimer's.
And they're bad news for brain cells.
Widely thought to be, yes. They're considered a major contributor to the nerve cell damage and death. The main forms studied are AB140 and AL142.
So being able to destabilize them or stop them forming would be a huge win.
Absolutely, a major goal.
And the second challenge, the blood-brain barrier, the BBB. Can you define that for us?
Right, the BBB, it's like the brain's bouncer, a highly selective protective layer separating the blood from the brain fluid.
Very strict gatekeeper.
Extremely. It keeps out toxins and pathogens, which is vital, but it also blocks most drugs from getting into the brain.
Making it a massive hurdle for treating brain diseases like Alzheimer's.
Exactly. So if you want to treat the brain effectively, you need a way past that barrier.
Okay, so the study tested these PLGA BPEG nanoparticles against both problems. What did they find?
The results were pretty positive, actually. First, the nanoparticles were very small, which generally helps with crossing biological barriers.
Is start.
And crucially, in the lab tests in vitro, they showed a destabilizing effect on the afibril formation.
So they could potentially reduce that harmful protein buildup.
That's the hope. And regarding the BBB, they successfully created a lab model of it.
An artificial barrier.
Uh-huh. And using that model, they showed the nanoparticles could cross it. And they released the Donepezil in a controlled way on the other side.
Wow, okay. Crossing the barrier and potentially tackling the fibrils? That sounds promising.
Definitely. But Alzheimer's isn't just about the protein clumps, is it? Inflammation plays a big role, too.
You're absolutely right. Neuroinflammation in the brain is increasingly seen as a key player in Alzheimer's progression.
And the study looked at this as well.
It did. They measured inflammatory molecules like interleukins and cytokines in their BBB model when brain support cells called astrocytes were exposed to those amyloid fibrils.
What happened? Did the fibrils trigger inflammation?
They did. Levels of several key inflammatory signals went up. Things like IL-1, IL-6, GM-CSF, TNF, IA, also TGF-AC and MCP-1.
Okay, hold on. Can you give us a quick rundown of what those are? IL-1, IL-6?
Sure. IL-1 and IL-6 are major pro-inflammatory interleukins, signals that ramp up inflammation. TNF-AT is another big pro-inflammatory cytokine.
Right. And GM-CSF.
That's a growth factor, but it can also stir up inflammation. MCP-1 is a chemokine. It basically calls other immune cells to the area.
And TGF-A.
TGF is a bit more complex. It can have both pro- and anti-inflammatory roles depending on the context. But overall, the picture was clear. The fibrils were causing an inflammatory response in these brain cells.
Okay, so the fibrils light the fire. How did the Donepezil affect that fire? Both the free drug and the nanoparticle version.
This was another really interesting finding. Both free Donepezil and the Donepezil delivered by the nanoparticles caused a significant decrease in several of those key inflammatory markers.
Which ones?
Specifically, IL-1, IL-6, GM-CSF, and TNF-F. The reduction was dose-dependent too, meaning more drug led to less inflammation. So it suggests a neuroprotective effect, calming down that inflammation.
That's the interpretation, yes. A potential anti-inflammatory benefit beyond its known effect on acetylcholine.
But not for all the markers. You mentioned TGF-F and MCP-1 earlier.
Correct. No significant changes were seen for TGF and MCP-1, which suggests Donepezil's anti-inflammatory action might be specific to certain pathways.
Fascinating. So Donepezil might be doing more than we thought, especially when delivered this way. Now, you mentioned PLGA isn't new.
No, not at all. PLGA has been a workhorse in nanomedicine for decades, actually. Used for drug delivery in many different diseases, not just Alzheimer's. Its biocompatibility and biodegradability make it really attractive.
And the research mentioned other nanostructures too, like polymersomes. What are they?
Right, polymersomes, think of them like synthetic bubbles. They have a bilayer membrane, similar to liposomes, which are made of fats.
But polymersomes aren't made of fat.
No, they're made from the self-assembly of special polymers called amphiphilic block copolymers. These have parts that like water and parts that hate water.
Okay.
So in water, they automatically form these stable, hollow spheres. They're often considered more robust than liposomes.
And useful for carrying drugs.
Very. They can carry drugs that dissolve in water and drugs that don't within their structure. Another useful tool in the box.
Definitely sounds like it. But back to PLGA. If it works so well in the lab, what's stopping it from being used widely now? Are there challenges in making large amounts?
Ah, yes. Scaling up, that's a major hurdle. Moving from small lab batches to industrial scale production isn't simple.
Why not?
Well, many lab techniques, like those using emulsions, are tricky to control perfectly when you make them much bigger. You can get variations in nanoparticle size, properties, drug release. Consistency is tough.
I see.
And even newer methods, like microfluidics using tiny channels, can have issues. PLGA can sometimes be sticky and clog up those tiny channels, especially if they're made of certain materials like PDMS.
So production bottlenecks. Are people working on solutions?
Oh, absolutely. Lots of work is going into improving manufacturing, developing better continuous processes, using different materials for those microfluidic chips like glass or Teflon, polytetrafluoroethylene that are less prone to clogging and can handle higher pressures.
Any breakthroughs mentioned?
The research highlighted one interesting new method for making PLGA microspheres a bit bigger than nanoparticles, using something called emulsification in a packed bed column. It apparently offers high yield and is pretty fast, which could be a big step for pilot-scale production.
Good to hear practical progress is being made. Now, once you have the nanoparticles, how do you know where they go in the body? Biodistribution studies, right?
Exactly. Biodistribution is critical. You need to track where the nanoparticles go, how long they stay there, how the drug gets released.
And PLGA helps with this.
One advantage is protecting the drug. PLGA can shield medication from being destroyed in the stomach, for instance, potentially increasing how much actually gets absorbed into the bloodstream. That's bioavailability.
But it's not perfect. The research mentioned a lack of specificity.
Right. A drawback of basic PLGA is that it doesn't inherently know to go to the brain, for example. It can spread out. And sometimes blood proteins stick to it.
And that's bad
Because the immune system can then recognize these protein-coated particles and clear them out, maybe before they even reach the brain target.
which brings us back to modifying them again, like adding that PEG layer we discussed.
Precisely. Pegylation helps reduce that protein binding and immune recognition, letting them circulate longer.
Any other modifications mentioned for brain delivery?
Yes, coating the PLGA nanoparticles with something called poloxamer 188 was mentioned as another strategy that might help them cross the blood-brain barrier more effectively.
So it's all about making these carriers smarter and stealthier. Now, we've focused a lot on Donepezil and PLGA, but is nanomedicine for Alzheimer's broader than that?
Oh, much broader. It's a really dynamic field. Researchers are exploring many other angles beyond just delivering existing drugs better.
Like what?
Well, one big area is interacting with the brain's own immune cells, the microglia.
Microglia, can you define those?
Sure, microglia are the resident immune defenders in the brain. They're supposed to clean up debris, like those amyloid beta proteins.
Through phagocytosis, eating the bad stuff.
Exactly, phagocytosis. But in Alzheimer's, their cleaning function can become impaired or even counterproductive.
So nanomedicine might try to boost them.
That's one idea. Develop nanoparticles that could enhance the microglia's ability to clear AP effectively, maybe without triggering too much harmful inflammation at the same time.
Interesting. Any specific examples of other nano approaches?
The sources mentioned one called NCKLVFF. It's a nanocomposite.
NCKLVFF. What's that made of?
It has a core of a common protein, bovine serum albumin, and attached to it is a specific peptide, a short chain of amino acids called KLVFF. This peptide is known to interact with amyloid beta.
And what does it do?
Lab studies suggest it can do a few things. Inhibit the early clumping, the oligomerization of ABRA, encourage microglia to take up and clear AF, and also help regulate certain receptors on microglia involved in that uptake process.
A multi-pronged attack at the nanoscale. Very cool.
Yeah, but despite all this progress, there are still major challenges, right?
Definitely. Big hurdles remain. One is standardization.
Yeah. Meaning there aren't yet universally agreed upon ways to make and test these nanomaterials. This makes it hard to compare results between labs and ensure quality control if things move towards clinical use.
Yeah, that sounds crucial.
And, while we know a lot more about Alzheimer's than we used to, it's still incredibly complex. We don't fully understand every single biological mechanism involved.
Which makes designing perfect therapies difficult.
Exactly. It's hard to hit a target perfectly if you don't have the complete map, but the potential is definitely there.
So wrapping this up,
Hmm?
what's the key takeaway for you, the learner, listening to all this?
Well, I think it's clear that nanotechnology, using things like these PLGA nanoparticles carrying drugs like Donepezil, offers a genuinely new and promising angle on tackling Alzheimer's.
The ability to potentially get past the blood-brain barrier, deliver drugs more effectively, and even reduce inflammation. These are significant steps.
For sure. And while challenges like scaling up production and fully understanding the disease are real,
The research really does underscore the incredible potential of these tiny tools.
it really does. Which leads to a final thought for you to consider. As this nanomedicine gets more sophisticated, what are the ethical considerations we need to think about, especially when intervening directly in the brain for diseases like Alzheimer's? Something to ponder.
A very important question for the future.
Thank you for joining us on this deep dive into bio bits and bytes.